Hello class, today I'm going to inform you guys about stem cells. So what are stem cells? They are unspecialized cells that have not yet decided what type of adult cell they will be. They are able to self-renew and become new stem cells. They can differentiate and become specialized cells. Some examples of this are blood cells, brain cells, heart muscle cells, and bone cells. So some different types of cells. First one are adult stem cells. Second, embryonic stem cells. Third, induced pluripotent stem cells. Why are stem cells important? Stem cells can be used to replace defective or dying cells, making them a source of potential new cures and therapies for numerous of chronic diseases or injuries such as diabetes type 1, heart disease, Parkinson's disease, and spinal injuries. Some therapies using stem cells. There's stem cell transplant. That is where the embryonic stem cells are first specialized into the necessary adult cell, and then the mature cells can replace damaged tissues. An example is a treatment like this can replace neurons damaged by spinal cord injuries, strokes, and Alzheimer's disease. Some stages in a stem cell procedure. The first stage are when stem cells move from bone marrow to bloodstream using chemotherapy and synthetic growth factor. Second, a machine collects blood and separates out the stem cells. The third is when the stem cells are frozen and ready to return to the body later. The fourth stage is where chemotherapy is used to fully, partially wipe out bone marrow and the immune system. Fifth stage, stem cells return to the body using a drip. And the sixth stage, the body recovers for a period of 3 to 6 months and sometimes even longer. So some pros and cons. Adult stem cells. Pros. Less likely to be rejected if used in transplants. Success has been demonstrated in clinical applications. Some cons: they cannot be grown for long periods of time in culture, and no current technology available, and there is no ethical concern with setting adult stem cells. Pros and cons of embryonic stem cells. Pros: they can maintain and grow for more than 1 year in culture, are established protocols for maintenance in culture. And cons: uncertainty, rejection during transplants, not a lot of testing/research has been done with embryonic stem cells, and to obtain the inner cell mass, the embryo must be destroyed. Pros and cons of induced pluripotent stem cells. Some pros are there's abundance of somatic cells of donor can be used, useful for drug development and developmental studies. Cons: has been shown to cause cancer in mouse studies and have the potential to become embryos if exposed to the right conditions. Some controversies with stem cell research. First, the disagreement with the definition of human embryonic stem cell. Second, the concern that derivers of stem cells might profit from the development of human embryonic stem cells. Third, they require that the IVF physician and HESC research should be different individuals to prevent conflict of interest. Fourth, concern about the monitoring of funded research and the invocation of penalties for research that do not follow the guidelines. Eligibility of human embryonic stem cells for research with NIH funding. Human embryonic stem cells should have been obtained from human embryos that were created using in vitro fertilization and no longer need them for reproductive purposes, that were donated by individuals who gave voluntarily consent for them to be used for research purposes. The following should be included: consent forms or other documentation upon donating human embryos. All options available in the health facility pertaining to the embryos no longer need for reproductive purposes. No payment or cash were offered for the donated embryos. Policies/procedures were in place, and that neither the refusal or consent of donating human embryos could affect the quality of care provided to the donor. And that is my speech about stem cells.